As we know, the organization and analysis of biological data are the main activities of bioinformatics. In the flow of genetic information from sequence to function, the stored information is translated twice. First from DNA to mRNA in the process of transcription, then from mRNA to protein in the process of translation. DNA and protein sequence comparisons have become routine steps in biochemical characterization from newly cloned proteins to entire genomes. Methodology used in the field of bioinformatics: sequence analysis, genome annotation, computational evolutionary biology, literature analysis, analysis of gene expression, analysis of regulation, analysis of protein expression, modeling biological systems, high throughput image analysis, molecular interaction, prediction of protein structure.
Of 8,000 kilometer and also exclusive
Result: biotechnology has played a major role in the areas of induction and control of maturation and spawning. Sex inversion in protandrous species like sea bass and protogynous species like the grouper, production of triploid, tetraploid and transgenic fishes. Traits that are being tested in fish species such as carp, trout, salmon and channel catfish include growth rates that are 3 to 11 times faster with more efficient feed utilization, increased tolerance to cold water and improved disease resistance. The final set of results explore how well the functionally equivalent species can predict future biomass and the underlying state for the geographical system.
or with any other developed
Conclusion: biotechnological research and development are growing at a very fast rate. The biotechnology has assumed greatest importance in recent years in the development of fisheries, agriculture, and human health.
alone
The science of biotechnology has endowed us with new tools and tremendous power to create novel genes
of
and genotypes of plants, animals, and fish. The application of biotechnology in the fisheries
fisheries existing
sector is a relatively recent practice.
mariculture
Nevertheless, it is a promising area to enhance fish production. The increased application of biotechnological tools can certainly revolutionize our fish farming besides its role in biodiversity conservation. The paper briefly reports the current progress and thrust areas in the transgenesis, chromosome engineering,
like
use of synthetic hormones in fish breeding, biotechnology in health management and gene banking. Seeing the potentiality of ornamental fish culture, emphasis may be given on systematic cataloging of commercially important aquarium fishes, bringing new ornamental fish species for diversification, detailed studies on breeding biology and behavior aspects, nutrition and feed formation, disease diagnosis and comprehensive health management, fabrication of aquarium associated equipments through education and training.
the ponds
Thank you all. Thank you for your patient listening. Thank you.
which are happen to be the
Nice presentation, Ms. Kirtana. Any queries?
separate assembly of organisms
Okay.
and
So
between
next, I would like to call upon Ms. Sune from Banari Amman Institute of Technology.
from land to freshwater,
Yes, ma'am.
then fresh
Yeah,
water
please share your
to
presentation.
the coastal water. And when we are in the coastal water, what exactly is the present status of the coastal water aquaculture already we have seen. But we did not venture into the real mariculture.
Is it visible,
Real
ma'am?
mariculture is there in the open sea,
Yes,
in the
it's
deep
visible.
sea.
Good evening to
One of the
everyone. I'm
challenges
here to present on the topic
is
aquaculture, nutrition and fish feed.
even if you just
I'm
take a
Sona from
test
the
of
Department of
freshwater
Food
aquaculture,
Technology, Bannari Amman Institute
it
of
started with the
Technology.
carp, and then
Aim and
after
objective.
the catfish is
Manufacture
also
feeds are an important
used,
part of modern commercial
and
aquaculture as it
aquaculturation
provides the
technique,
balanced
which with the
nutrition needed by
artificially
farmed fishes.
produced the seeds, revolutionize the thing.
Methodology.
And
These are
this
the important
is the one
component of
important
whole
reason
production
why
system.
even today
Overfeed,
we can get in the market
overfeed and underfeed are the
the
main
fish
components,
for about
and
50 to 60
growth,
rupees a
FCR,
kg.
water quality, danger,
If you
pond
see
protection,
many of
closed
the vegetables are
recirculation
much higher
systems
price
are
than
the
this.
major components
But
other than
the kind of
overfeed and
protein
underfeed.
which the fish provide is much superior to even the vegetables.
Fish
So
requires food
this is
because
how
it
we
needs
could say
energy
that
to do most of its movements and
there is
all other
a
engagements.
revolution in the
And
fish
the aquatic
aquaculture,
animals are cold-blooded,
and
but they do
this
not
has
differ
led
from
to
farm
the
animals
blooming
and
of
humans. Protein, fat, carbohydrates, vitamins, and minerals are the essential ingredients.
When we think about this aspect,
Small fish requires more protein than larger fish, and the proteins need to be of good quality.
marine fishes are much better than freshwater fishes. Marine fishes are much better than even the organisms or animals formed
Even
on
though
animals
carbohydrates,
formed on the
even though carbohydrates are the main components of fish food,
land. You
it
take
is not
poultry or you take
not much
the
research are done
pigs
on
or
carbohydrates,
piggery
and
or
it has
you take
limited
the
digestibility of
cattle
35 to
farming.
80%.
All these farming
Cobalt, chromium, copper, iodine, iron,
feed
manganese,
takes
selenium,
a
and
less
zinc
quantity of the
are
fish.
included in microminerals.
For
These
example, in the case of
microminerals are also known as trace minerals.
pigs, it is 4 kg. In the case of the cattle, it is around 10 kg. 10 kg of feed can be
Vitamins
converted to
cannot be
only 1
synthesized,
kg
but
of meat.
it must be
While in
supplied.
the case of
And there
the
are two types
fishes,
of
it is
vitamins: water
1.2
soluble and fat
kgs of
soluble.
feed which can be converted to 1 kg of
Since
fish
aquaculture
flesh.
is an emerging field and it is making significant change in
This is the
economics of
very
India, it is
important point.
important to feed the fish with proper
So
nutrition and
more
health
than the
care.
land
So I conclude,
segregated
it is
organism,
important for every
that
aquaculture
even
growers
with
to
regard to
maintain
the
nutritional balance
freshwater
in
fish,
fish feed. Thank you.
with regard to the freshwater fish,
Thank you,
it
Ms.
is
Sona.
much more important to
Any
culture
questions?
marine fish. And what is the reason?
So we have the last
Marine
presenter,
fish,
Ms.
in the case of the
Philomena
freshwater
from
fish,
Institute of
what happens
Oceanography,
is that there
University of
which
Calabar,
require more
Nigeria.
metabolic processes. But in
Ms.
marine
Philomena,
fishes, it is
please
much
share
more
your
less.
presentation.
Even with regard to the land fishes, in the land organisms like sheep or goat, most of their energy goes into the production of the hardy skeletal material like bones. That is why
Ms. Philomena,
there we have less
you there?
production. The fish has got the minimum bare required of requirement of the bones. That is why more flesh is there. Further, there is no gravitational pull for which quite a lot of energy has to be spent. With regard to the marine fishes, it is a very ideal condition, no climatic drastic
Philomena,
changes.
please unmute
So
your mic.
everything is meant for it. Probably this medium, if we just try to look at the saltwater medium, where the life is originated, it is something very, very, uh, what do you call, ideal
Philomena?
medium for the life to sustain. So
She is
from
there,
that
she
point
has
of view,
come, but
Yes, sir.
her mic is.
Please unmute
we
your
have
mic.
started culturing the organisms and we could find some solutions. So once when we have vented to the land to freshwater, freshwater to the coastal, and this entering into the marine fishes not ventured so far, what could be the reasons? There are quite a lot of things. For example, if we take the infrastructure, the regulatory acts, the kind of feeds available, all with regard to all these things, there is immense problem in mariculture. In the case of the freshwater fishes, the size of the egg, fish egg, is much bigger compared to that of the marine fishes. As a result, we cannot culture them easily, or we cannot produce their seeds in the hatchery the way in which we produce the seeds of either catfish or the
Yes, we can
or
see your
the seeds
screen.
of
Please
freshwater fish.
unmute your mic.
Nevertheless, if we consider the economic point of view, if a catfish or
Voice is
Are you
a
seeing our
cat
screen?
will give you only
Yes.
50 to 70 rupees per
Can you
kg,
see my screen?
while marine fish will give
Now
700 to 1000
we
rupees per
can
kg.
hear
Can
you.
I see?
There is a great demand for
Okay,
tropical fishes, offshore ponds
We
and
can hear
aquariums.
your voice
Even
and your screen is sharing.
in the
Please
80s and
share your
can
90s,
you
screen.
see the
the
presentation?
crab or shrimp, particularly the penne
Hello?
mignon, has been sold for 700 to 800
Hello?
rupees on the
Yes,
value
we
Yes,
can hear
please.
you.
so that is the
So please,
advantage of
my
the
student is
marine
going to
culture or
make a
the
presentation now, my PhD
aquaculture.
student.
And
Honor
this
Stagius. Please listen.
this
Go
culture,
ahead.
mariculture, has
The
gained
topic is
some
growth and hematological status of Clarias
and
garapinus
first
fed
characteristics.
Jatropha
What
tagorensis
are
leaf
the
powder meal.
seeds?
Aim and objectives.
And this
To
availability
determine the effect
has
of
been in
Jatropha
vitro
tagorensis
research.
leaf meal
There have been
on the hematology of Clarias garapinus fingerlings to determine the growth rate of Clarias garapinus fingerlings fed with the meal to estimate safe dosage of JLM in feed of Clarias garapinus fingerlings. Methodology. Fresh leaves of the plant were obtained from Botanical Garden, University of Calabar, Nigeria. Plant leaves were processed in the laboratory to powder form as recommended by Musso et al. 2000. 75 healthy and acclimatized fingerlings weighing between 3.5 and 4.5 grams were stocked in 5 indoor plastic tanks at a density of 5 fingerlings per tank in triplicate. They were fed at libitum with different graded levels of experimental meal at 0%, 10%, 20%, 30%, 40% for 98 days. Basic water quality variables were maintained at their optimal levels throughout the experiment. Growth indices such as weight gain, feed conversion ratio, specific growth rate, and survival rate were determined as demonstrated by Agbebi et al. 2012. Blood samples were collected with syringe and preserved in sodium salt of ethylene diamine tetraacetate acid for hematological analysis. Hemoglobin was determined with the cyanohemoglobin method. Fat cell volume was determined with the microhematocrit method. Red blood cells was determined with the improved laboratorial cytometer. White blood cells was determined with the improved Nabura counter. Statistical analysis. Data obtained from the experiment were expressed as mean plus or minus standard deviation using one-way analysis of data. An acceptable value of p less than 0.05 was considered to be statistically significant. Results. Growth response. At the end of the 96-day experimental period, results showed that highest mean weight gain as 363 grams was obtained from fish-fed 30% plant meal, while least 200.1 grams was obtained from those fed 0% plant meal, as given in the figure. Similarly, specific growth rate was highest in 30% group and lowest in 0% group. Feed conversion rate.
Hello.
Yes, please.
Okay, communicate.
We can hear you properly. Please proceed.
Okay, go ahead.
Results. Growth response. At the end of the 96-day experimental period, results showed that highest mean weight gain, that is 363 grams, was obtained from fish-fed 30% geographic leaf meal, while least 201.1 grams was obtained from those fed 0% plant meal, given in the figure 1. Similarly, specific growth rate was highest in 30% group and lowest in 0% group. Feed conversion rate ranged from 0.8 to 1.50, while mortality rates ranged from 5.0% in 30% group to 9.33% in 0% group. Hematological status. Hematological analysis revealed that packed cell volume was highest in 30% group and lowest in 0% group. Red blood cells was highest in 30% group and lowest in 10% group. White blood cell was highest in 30% group and lowest in 0% group. Hemoglobin was highest in 30% group and lowest in 0% group. Our figures was modeled and we saw that Clarias garapinus fingerlings was dose and time dependent according to the growth, while hematological variables were dose rather than time dependent. Diet with 30% of plant meal gave the best performance in growth and hematological responses. So we concluded that Clarias garapinus fingerlings can thrive best when fed with 30% inclusion level of the plant meal. Thank you.
Thank you, Philomena. Nice presentation.
Yeah.
Any queries from the participants?
a nice presentation, and with regard to the fingerlings, growth and survival, she has given a very good account. Thank you for communicating to us and presenting your paper in this conference. We are very happy.
Thank you very much.
Thank you very much.
We are grateful for the opportunity.
It's nice. It's our pleasure.
Fine, sir. We are done with the oral presentation. Sir, I'll move forward. We are done with the oral presentation.
Sure, please.
As we have come to the end of the program, now I request Professor Dr. K. Altaf, sir, convener, to deliver the valedictory address and give concluding remarks. Over to you, sir.
Good afternoon to all.
Ms. Philomena, please stop sharing your screen. And unmute your mic.
Thank you, sir. Please, sir.
Ms. Philomena, can you please unmute your mic?
Okay.
Please, sir.
Good afternoon to all. I think it is one of the fruitful days we had as far as getting information about aquaculture. In fact, this program started with a valid lecture on aquaculture by our respected registrar, Jayaprakash Vail, where he has mentioned about the importance of the food security and need for alleviating the protein deficiency. And it is followed by the first session, invited lecture. It is by Dr. Kailasam, CSIR SIBA scientist, a person who has done quite a lot of work on fish broodstock development and also their propagation and various technologies the SIBA has used along with Dr. Arasu, and who is now retired, but both of them has contributed a lot. And in this lecture, he has shown the number of fishes which have been domesticated and also seed production and hatchery development, as well as farming technologies. So different fish requires different technology to be adopted, and which has been propagated to almost all the coastal states, right from Gujarat up to West Bengal. So this has enhanced the economic state of the people, as this mostly these fish SIBAs being a euryhaline, it can be cultivated even in some of the inland ponds, but at the very same time it fetches a good market price also. So it is a wonderful lecture we had, and the second section we had from Dr. Shaul Hamid, Sheikh Shaul Hamid. Again, say we have such a genius person among us today. We had, he is somebody who has established the first the virus identification kit in India, and it has been now propagated all over the world. And such a eminent person has given even the minute details of identification of a virus, his ultrastructure, the kind of protein constitution, and the RT-PCR techniques in detail. And finally, he also said, if it is not just the diagnosis of the virus, but also finding a solution. And he has made it through the herbal products which have been addressed to this issue. And as a result of that product development, with the help of the DBT, there is a great relief for for the shrimp farmers. Now, venom can be cultivated at higher concentration or higher quantity. So after this, again, we had our Dr. Santanam, who is again a very, very important person, because the field on which he is working is very important. Being a copypodologist for the past 40 to 48 years, I am living with this organism. I know how difficult it is to grow them, understand them. It is not like Artemia or the rotifers, because Artemia and rotifers happen to be parthenogenetically developing organism. If you give food, they will grow, but copepods are not like that. Why copepods are superior? They are superior because they are sexually reproducing. That means there is not going to be any dilution in the quality of their nutrients. And that it is not easy for them. Please remember, if a copepod has to produce eggs, the male has to transfer the spermatophore to the female. The size of the spermatophore is one-third of the length of the copepod. In water, suspended condition, in high density, with so much organism around, how it is possible to do that? So we need to understand all those things and try to formulate the protocol for high density production. In that, Dr. Santanam has achieved it, and he is also going to the next stage where he is going to commercialize the product. That is a very, very welcome aspect, because if we have suitable live feed, the number of shellfish and also the finfish species production can be uh, escalated. The upscaling of the finfish farming will be something which will be quite beneficial, because there is quite a lot of demand from the Western countries for this fish meat. So that way, that is very important. In the same tune, the importance of the algae and what exactly are the different diversity of the algae, and what kind of wealth God has bestowed to our waters has been very lucidly illustrated with a very good pictures by Dr. Mayakanan, who has mentioned the various advantages of this algae, like pharmaceutical, medical, nutrient, nutraceuticals, and in so many different ways. So somebody who is now, say we were having a different opinion about the algae. Always when there is a bloom and it is going to be something drastically crash and then spoil the system. But I think his lecture might have now infused a kind of confidence in the youngsters, where we can take up such things and try to convert which we think as the waste into something gold. So such is the lecture by Dr. Mayakanan and Dr. Edward Danish. When we have all infrastructure, all facilities, and mind to work, animals are there, everything is there. But if we don't concentrate on the biosecurity, then everything is going to go waste. So in a very, very systematic way, in a detailed manner, he has enumerated, he has informed about this, what kind of biosecurity level testing and retesting, and making sure we procure the hassle-free seeds, healthy seeds, quality seeds, and without any kind of contamination, we are growing them and trying to harvest and get benefit out of them. If we don't do that way, then the entire investment is going to waste. What we have to do, simply shut down the farm or shut down the hatchery. This is what it is going to be. So that way, again, Dr. Edward has given a wonderful lecture. And coming to the paper presentation, I'm very happy. I'm too happy. I always make a provision for the budding scientist. And today we had presentations. Of course, we had more number of papers, but due to some technical issues, that is not availability of the requisite facility for them to present it. And also because of some other issues like pandemic, we could not accommodate everybody. It's not possible for everybody to present. Anyhow, we had at least a dozen of presentation where from basic science to advanced molecular studies up to that level, the papers have been discussed. In overall, I hope the participants' mind might have been mm, fully saturated with the aspects of the mariculture or aquaculture and what kind of things we have and how exactly we should uh, proceed further, at least for the young scientists, the young staff members, research scholars, PG students. They should take many things from today's conference. It is something, if you have a confidence, there is funding agency like DBT, DST, UGC, and so many other ICR, ICMR, everything is there to give you money if you have a real original idea and also a willpower to design something and try to do something innovative. That way, this conference might have, I think, benefited all the participants. In this regard, I have the pleasure of thanking our management, our chancellor, our pro-chancellor, our vice-chancellor, our registrar, COE. They always they promote such activities, and they are always one thing in their mind is we should produce good citizens, we should produce skilled degree holders, and also their aim is we should do something for the benefit of the country. So for towards that, our conference is something which is really has contributed a bit. And also, I would like to thank the Indian Association of Aquatic Biologists. I am very, very thankful to Dr. Mulay, who is the president of this association, Dr. Shama Madam, who is the secretary, and also Dr. Salaskar, who is Bombay-based. He is a limnologist. And this association is publishing a journal, Journal of Aquatic Biology, and also is doing quite a lot of good services by publishing books with regard to the identification of the local fauna and flora, aquatic fauna and flora, as well as even trying to produce the kits and also distributing the water testing kits and other things in a actually a product, a subsidized kits of taking the help of the corporates. So that way, the Indian Association of Aquatic Biologists is doing a wonderful service to preserve the freshwater bodies, riverine system, lakes, and all other water bodies. Only if those water bodies are clean and pristine, it is not going to pollute the ocean. Otherwise, ocean will get polluted. That way, we have really we are thankful to the Indian Association of Aquatic Biologists joining us in organizing this event. And I thank all the members of the organizing committee, Dr. Sendilnathan, the head of the department, Dr. Selvakumar, Dr. Mayakannan, Mr. Shortly is going to become Dr. R. Vijayaraj, and again Ms. Sorna, she is also going to be Dr. Sorna in a few days, a few months. All have submitted their thesis, and Sudha, who is in the verge of submission of the thesis, and all other scholars of the AMET University, the PG students, everybody contributed their might towards making this function a grand success. And with this, I thank all the participants, all the presenters, all the participants for responding to our call and making this function a grand success. Thank you very much.
Thank you, sir. I now request Dr. Senthilnathan, sir, Head of the Department, Marine Biotechnology, to propose vote of thanks. Over to you, sir.
Thank you, Sana. So, We are the last session. Good evening to one and all. I take the privilege of proposing the vote of thanks in the wonderful evening to the cheerful participants. My hearty thanks to our Vice Chancellor, Colonel Dr. G. Srivastava sir, the man of spirit, as I always mention. His actions are always expressed that the age of mere numbers. He is very dynamic and a serious taskmaster. My sincere thanks to our beloved registrar, Dr. M. Jayaprakash sir, for his untiring support, necessary arrangements for this conference, and man behind the show. It's my great honor to express my heartfelt gratitude to the guest speakers of the conference. Dr. M. Kailasam sir, Principal Scientist, SIBA, has presented the current status of aquaculture industries in India, especially with its marketing potential that motivates all of us to think about the aqua farming and be an entrepreneur. Thank you, sir. Second, Dr. A. Savlamit, sir, is an expert member in aquaculture industry. He has highlighted on the importance of disease diagnosis, proper health monitoring of the streams, and development of successful products. Thank you, sir. Third, Sandanam, sir, from Bharatdasan University, Trichy, as illustrating the sustainable mariculture and the industrial scopes of the coffee flocks. Thank you, sir. Fourth, by Edward Danisher, Head Production, Business Development and Research, GKHS, for highlighting the hatcheries design and infrastructure, do's and don'ts, and maintenance of the aqua farms. Thank you, sir. Very crisp and sharp presentation. And fifth is by my colleague, Dr. G. Mayakan, sir. His talks is kindling the interest on all the research, production, and marketing scopes. Thank you. Thank you for all for accepting our invitation, and we feel so honored to have all your talks in the conference, which is definitely creating an insight to the listeners. My hearty thanks to our godfather, Professor Dr. K. Altaf, sir, my colleagues, Dr. Selvakumar, and Swarnakala and Vijayaraj, and all other scholars, IT support team, PRO department for designing the brochures and invitation. I chiefly thank the deans, heads of the various departments, directors, and media. I cordially thank the stars of this conference, so it's none other than the participants. The participants, those who are making this event to be a successful one, thanks to the digital platforms that makes easier to connect the people across the globe. Since we are making this is a national conference, one student presentation was made from African countries. So, and that makes the event international. So thanks to all your contributions. I also express my sincere thanks to Dr. K. Sharma, Secretary of Indian Association of Aquatic Biologists, and Dr. Salaskar for associating with AMET University in the conduct of this conference. Once again, I thank all the dignitaries, those who are all attending today. Thank you all for giving me this opportunity. We meet you all in the next year. Thank you. Thank you for being with us and supporting us. Yeah.
Thank you, Dr. Sendil, for
Yes, sir.
making it great. Swarna, you can announce so that certificates will be
Yes, sir.
issued in due course.
Yes, sir.
And
I
also
kindly
the
request the participants. Yes,
abstract
sir.
also, feedback also.
I kindly request the participant to fill the feedback form. The link is in the comment box. The certificates will be generated after filling the feedback form. Now we'll start the first session, and I feel privileged to introduce our first speaker, Dr. M. Kailasham, Principal Scientist,
So regarding the abstract,
from Central Institute of Brackish Water Aquaculture,
Yeah.
Chennai. He has done his PhD
Sir,
from
I think we have
regarding the
received all the
abstract,
abstracts.
in marine science.
Yes.
He has
Abstracts
27
are
years of
ready.
experience
We will be
in the
distributing it with certificates to them.
and live feed culture. He has completed 7 major research
The
projects
participants
and
are
4
encouraged to
consultancy
submit their
projects
full-length
from
articles
venture
also.
funding
Yes.
agencies. And also, he has received 12 awards
We are
The
running a
procedure
journal.
we
Yes,
will
sir.
be processing the papers and then publishing it in the form of a proceedings. So we request all the uh,
during his
invited lecture
research period, he has
others as
developed
well as those students who have those scholars or staff who have presented the papers to submit a full-length paper by the end of this month, so that it can be processed and published in the form of a proceeding of the conference.
We are publishing all the articles to the ISBN number proceedings, so please
For the
submit
full
your
paper.
support. Yes, sir. Thank you, sir.
Thank you. Thank you.
Thank you, Sona and Vijay,
Thank you, sir.
for the all back and works and front end work.
Yeah, yeah. They are the backbone of
Yeah.
many things. So, thank you, Vijay. Thank you, Sanna.
Thank you, sir. All teamwork, sir.
Yes.